welcome, and we are ready to listen to you.
Good night, and thank you so much, Professor Ali, for the very warm welcome, and thank you all for coming this morning. So, we're going to talk a little bit this morning about large-scale brain imaging in psychiatry, and how can large-scale brain imaging studies reveal insight about mental illness. And this is work from a large-scale international project called ENIGMA. So if you are imaging the brain, there are several methods available to do it. You can use MRI to look at the structure of the brain. You can use positron emission tomography, PET, to look at the molecular content of the brain. There's a technique called optical intrinsic signal imaging that's used surgically to look at the brain's activity. And then there's all the classical neuroscience techniques that have been used to look at the brain's connections and cells. And we're interested in how our human brains change throughout life as we develop and as we age. So about 10 years ago, we started a project called the ENIGMA Consortium, which has published the largest neuroimaging studies of 15 brain disorders. The goal is to understand which factors help or harm the brain by analyzing brain scans and genetic data from 100,000 people from 45 countries. And this is work by 2,000 scientists in 51 different working groups. And it has led to the largest neuroimaging studies of several diseases: epilepsy, Parkinson's disease, ataxia, and in psychiatry, bipolar disorder, major depression, schizophrenia, and in brain development, OCD, autism, ADHD. So the main goal of ENIGMA in analyzing 100,000 brain scans is to ask, how do 33 major disorders affect the brain using imaging methods, MRI, DTI, resting state fMRI, EEG, and MEG? And also, how does our genetic variation affect the brain? So we screen millions of markers across the genome with different techniques to find genetic markers and environmental factors that affect brain development and aging, disease risk, and treatment response. So there are three reasons to collect data from across the world. One is statistical power. A large amount of data is needed to find connections between the genome and the brain. Efficiency. So a lot of distributed computing across the world can be used to process the images. And team science, the collective intelligence of a large number of people, we benefit from that. So today's talk, I'll say a bit about what is ENIGMA and how do neuroscientists from different countries work together. We'll talk about a worldwide genetic analysis of brain images, how to link neuroimaging with genomics. We'll talk about some global studies of brain diseases and some challenges and advantages of pooling data internationally, and then finally some fun new directions with artificial intelligence, AI, and so-called deep learning with brain images. So here is the data. So all of these sites around the world are collecting brain scans, brain MRI, and the radiology images are located at these centers. They're not transferred to any one place. And the color is the main disease that that hospital is studying, and there are maybe 30 or 40 diseases. This approach has been reviewed in different journals, Science, Neuron, as a method to collectively analyze brain images. And the work is divided into many different clinical groups. So as you see here, some groups study bipolar disorder, schizophrenia, major depression. There's other groups that study early onset psychosis, schizotypy, and clinical high risk. In neurology, there are groups studying stroke, epilepsy, ataxia, Parkinson's disease. And then mainly in children, there are groups studying obsessive compulsive disorder, autism, ADHD, and Tourette's syndrome. Now, each disease or disorder is not homogeneous. And so if you have a brain injury, you may have the injury in the military, or in sports, a head injury, or children. And there are also very sad cases in intimate partner violence, where in domestic abuse or child abuse there is a brain injury. So if you study the imaging from these conditions, sometimes they're studied separately because the etiology is different. Also, methods groups. These methods for imaging, EEG, fMRI, diffusion MRI, there are non-clinical groups that provide infrastructure and support for analyzing data of these different kinds. So here's all of the different groups. There are psychiatric conditions, neurological conditions, anxiety, panic disorder, specific phobia. These are different conditions where each lab can only collect a small amount of data, but overall, when the data is put together, we can begin to see consistent patterns. So here are some of the people who run the projects. They're in different countries. And if you think of each lab, I mean, each dot is maybe a lab, and this is a collaboration between different labs. There might be laboratories that study one disease with MRI, diffusion imaging, functional MRI, and labs that study a different disease, disease 2 and disease 3. And there are all sorts of connections and collaborations between the scientists studying one disease with different methods or studying different diseases with the same method. So this has led to detailed pictures of structural differences in the brain in different diseases: major depression, bipolar disorder, schizophrenia, alcohol use disorder. These images and the scale represent the abnormalities in the brain's gray matter. So the brain's cortical surface shows thinning or reduction in each disorder, except for this one, autism spectrum disorder. In autism, the patients have increased gray matter. We don't entirely know why, but there is an increase, whereas in epilepsy, and this one is a deletion syndrome, a neurogenetic syndrome, 22q11.2 deletion syndrome, which is a deletion of genomic material that increases risk for schizophrenia in the people that carry the deletion. And you can buy a t-shirt that shows the data. So let's talk about how the images are processed. If we collect a brain scan from MRI, and if we collect a brain scan from PET, each lab can register or align the original scan with an atlas. So this Telerak atlas has been used in the early days of brain mapping to provide a coordinate system in which to report or average or do statistics on data across different subjects. If we try this in Alzheimer's disease, you could collect a brain MRI scan of a patient. You could classify the gray matter of the brain, the white matter of the brain, the cerebrospinal fluid, and you could ask, where in the brain is there an abnormality in Alzheimer's disease? The amount of gray matter in a patient with Alzheimer's disease is less by maybe 10%, 15% on average. This is the percent. This is the statistical significance. Mainly the temporal lobe is losing gray matter early in Alzheimer's disease. And this matches the accumulation of amyloid beta protein, plaques that form in the brain. So this is post-mortem. This is a living patient scanned with MRI. Now, if you were to watch a patient with Alzheimer's disease over a period of years, you see the ventricles get larger, but the cortical surface, it's very difficult to tell where the gray matter is changing. And even if you look in axial, sagittal, coronal views of the brain, the main thing in Alzheimer's you see is this ventricular enlargement. Hippocampal volume is slightly less. But you'll see how difficult it is to notice a pattern in the images. So one way that has been used to map the disease is to render the cortical surface, measure the thickness of gray matter at each point, and then when comparing patients and controls, you can say over a period of, in this case, 2 years, does the gray matter loss stay stationary? Does it spread? And in Alzheimer's disease, we see the limbic system, the temporal lobe of the brain, all of these areas show a progressive gray matter loss. This also happens in schizophrenia. So in schizophrenia, there's also a pattern of initially temporoparietal gray matter reduction, but also frontal gray matter reduction. These patients are very young. They have the disease in 12 years old, 13 years old, early adolescence. And you see this progression of gray matter loss compared to controls. In a drug trial, you can ask, does olanzapine, a second-generation antipsychotic, do patients with this drug show different patterns of gray matter loss? So this is an older drug, haloperidol, a first-generation antipsychotic. And the people who were taking this medication had an annualized loss rate, a loss rate of tissue early in the disease that was quite high. We don't think that this continues for more than about 1 year, but the olanzapine-treated patients, it was discovered that it slows brain tissue loss, at least in the chronic phase. One thing to remember is that normally a healthy person will lose gray matter. So in this case, we see that a healthy child loses gray matter initially in the sensory motor areas of the brain, proceeding to the temporoparietal areas, language areas, and into the frontal cortex. So in the end, there are also differences between a young child and an adult, and all of these processes can be modeled statistically by collecting scans at different ages. So suppose you were to look at brain metrics from many people, actually from many countries, over the lifespan. This is age 20 years old, 40 years old, 60 years old, 80 years old. The brain grows in childhood until the age of about 20, and then there's a gradual loss of brain tissue. And then after age about 60, there's a more rapid loss of brain tissue. But even at a given age, let's say you are 30 years old, there's a huge range of variation in the volume or the size of structures. This is not the whole brain. It's a part of the brain, the right thalamus. It's a nucleus of the brain. And you can see we're interested in understanding what is it that causes this profile of development, of steady loss, and of rapid loss. Is it genetics? Is it environment? Is it disease? So the data can be collected from different centers. Each color represents scans that were collected at a certain hospital. So sometimes a hospital only scans very young children, here 5, 6 years old. Other hospitals only scan elderly people. And so we try to make a composite, an overall aggregate picture of the changes going on. This has led to reference charts. So these structures of the brain, the caudate, putamen, thalamus, ventricles, they each have a distinctive profile of development and aging. So the hippocampus, rapid growth, slow loss, rapid loss. The amygdala is more stable. There's not much change between the age of 20 and 40. And then the thalamus, we don't think that this is true. We think that patients who are 80 or 90 years old, who are able to be scanned, are very unusual. And so if a person is 90 and in the scanner, they maybe have a better brain than the people who are 80. So you could use this as a reference. In women, if you look at the left hippocampus, you could say, what is the 90th percentile or the 10th percentile? And if a patient is scanned, we could find out where are they in this distribution. Are they not developing rapidly? Do they have excess brain tissue? Or is the rate of loss too fast for a person of that age? Some genetic variants have been found that cause people to lose brain tissue faster. So this gene 1% of us carry a genetic variant in TREM2. It's a risk gene for Alzheimer's disease, and we find that the temporal lobe areas of the brain in carriers of this gene show very rapid loss of tissue. This is useful for drug trial enrichment. It's interesting to enroll people who are likely to decline faster and see if particular treatments work for, in this case, Alzheimer's disease, because this is a gene that increases the risk by a factor of 2 for Alzheimer's disease. So this has led to interest in discovering how do we find genes that help or harm the brain. So normally, if a patient comes to the clinic, you could collect a brain scan. You could also collect a saliva sample to sequence the DNA. But making a connection between genetic variation and diagnosis, it may require 50,000 to 100,000 people. So one idea is that gene variants may affect brain measures more directly. Brain measures or biomarkers might relate to disease status, of course, but they may be more precisely and reproducibly measured. And this is the so-called endophenotype hypothesis or biomarker hypothesis, that if we were to collect images, we would be able to make a direct connection between the processes going on in the disease and the genetic drivers of those processes. Now, some people disagree with this. So some people say, if you want to discover genes that affect the brain, you'll need brain images from 50,000 people. Just because images are more expensive to collect than measures of height or weight, it doesn't change the power calculations. So Nick Martin is a very famous geneticist. He was the president of the Behavioral Genetics Association. And he did not believe that biomarkers would be more easy to study than diagnosis. He said it will need a lot of people. In fact, many people have brain scans and DNA. So this study in Nature examined the genetics of brain in 30,000 people. So we measured the volume of caudate, putamen, hippocampus, and we asked, what are the genetic variations along the genome that are associated with the volume of the structures on brain MRI? And for each structure, the hippocampus, the amygdala, the caudate, it's slightly different. So just to explain how it is done, we would measure the patients in a certain trait, in this case, the size of the hippocampus. We would sequence the DNA and find what is the nucleotide in each person. And then we would run a statistical test to see if there's a link between the genetic variant and the brain measure. And maybe people with this genotype have a smaller brain. Maybe people with this genotype and this genotype have a larger brain volume. And this is volume, but it could also be a pathology. It could also be the function or the molecular level on spectroscopy. Now, this can be done, a genome-wide association study, or GWAS. This can be done to identify genetic variants that are linked with a disease. So in Alzheimer's disease, you can ask, in patients with Alzheimer's disease, is there any consistent difference in the genetic code? In Alzheimer's disease, the ApoE gene is altered such that there is 3 to 15 times the risk for the disease if you carry a certain variant of ApoE. And that variant of ApoE is present in 25% of us. Very, very important gene, 25% of us. These other genes affect the risk for Alzheimer's disease by 10 to 20%. This was discovered in 2009, and since then, it has been discovered that 6% of the risk for Alzheimer's disease comes from ApoE, 2% from the other known risk loci, these, And 25% is still hidden heritability. We don't know where the genetic risk is coming from. But your annual risk of developing Alzheimer's disease, this is age, and this is predicted age-specific annualized incidence. There's a huge difference between people in the high percentile of genetic risk, maybe 20% per year incidence at the age of 75. So you can see that these genetic variants affect disease risk, in this case for Alzheimer's disease. Yesterday, Professor Robin Murray mentioned in schizophrenia we also have genome-wide association studies. The main hit is in an immune system region, chromosome 6, the C4a complement factor. Another important gene for schizophrenia is in the dopamine receptor D2 subunit. It's involved in dopamine neurotransmission. And now, in this huge study, there are, in fact, I think, well, let's look at this. This is the growing size of the schizophrenia GWAS. There are now 200 genetic markers that have been linked with risk for schizophrenia. And as time went by, more and more genetic markers have been linked with the disorder. What do ENIGMA members do with their scans? So, you can compute brain measures from the scans. In MRI, you can measure the cortical and subcortical volumes with the free SOFA. In diffusion tensor imaging, you can measure the microstructure of the white matter. But then, as before, you can test associations between brain measures and 1 million single nucleotide polymorphisms. And you can do a meta-analysis. You can combine effects across sites, and each site's vote depends on the sample size. So 6 genomic loci are associated with the volume of the hippocampus. This is the hippocampus. This is the front of the hippocampus. It was found that the ApoE region, actually, I'll talk in a minute about ApoE. This is a different region that affects hippocampal volume. It has a gene called harakiri, a Japanese word for death, suicide. The harakiri gene is involved in apoptosis, cell apoptosis. And it's thought that many of us have a variant in this gene that eliminates cells in this area of the hippocampus. A geneticist working in Dagestan, this is the high mountains of the Caucasus, the Kafkas region. Kazima Bulaeva is a Russian geneticist, and she is the doctor who treats patients who are living in the high mountains of Dagestan. And she found that this same gene, harakiri gene, which alters the volume of the hippocampus, she found that in her patients, it was associated with unspecific mental retardation. She found that there was linkage of genomics in this same region, genomic variants, in a genetic isolate. And this is a region where these people have a high rate of consanguinity. They have inbred marriage because it's very isolated. And she was able to discover the function of the gene in this area that removes hippocampal volume and also in these people causes mental retardation. Now, there are 4 novel loci associated with hippocampal volume. What about the Alzheimer's disease risk from ApoE? We know that ApoE4, 25% of us carry this gene. It's noticed that as you get older, people who carry this gene have a loss of hippocampal volume. By the time you are 60 or 70 years old, if you carry this gene, your hippocampal volume is 2 standard deviations smaller than when you were younger. And so this is a very clear age-dependent mechanism of ApoE, in the brain that we can map to hippocampal volume. Each part of the brain has a different genetic association pattern. So we find that the hippocampus, there's certain genetic loci that affect it, the overall intracranial volume, certain genetic loci. Is there anything interesting? Well, this one is interesting. I don't know if anyone knows what that gene is. It's the tau gene, so microtubule-associated protein tau. This gene is really important for moving organelles around cells. In Parkinson's disease, ALS, Alzheimer's disease, people that have a common variant in the tau gene have a very rapid pathology. And here is the Parkinson's disease GWAS. Two big signals, disruptions in tau plaques and tangles form in the brain in patients with Parkinson's disease. And as Professor Robin Murray noticed yesterday, this gene is involved in dopamine release. So in Parkinson's disease, we know that people have motor rigidity, difficulty initiating movement. And in the basal ganglia, dopamine is also involved in the initiation of movement. And there seems to be a problem in the synuclein alpha gene that's involved in synaptic vesicle release of dopamine. So a connection between schizophrenia and Parkinson's disease in this area. So you can carry on. You can look at the genetic architecture of subcortical structures in many, many people. I think the surprise is that the genetic program for each brain region seems to be a little bit different. Maybe not entirely surprising, but this is found. So this last month we have found 500 loci affecting the subcortical volumes, different for the caudate, for the hippocampus. And you could add up a polygenic score. So just like in disease genetics, you can say how much difference does it make to carry these genetic markers? How much difference to my disease risk? You could say how much difference does it make to the volume of the hippocampus? And 5% of the variance is explained for 4 key brain structures. Here they are: hippocampus, brainstem, putamen, caudate, based on a measure just from your DNA. Now, you could say, well, 5% is not much. It doesn't seem like a lot. But as we know, our whole life experience affects the hippocampus. So it's interesting that this is true. In non-European people, people of non-European ancestry, we don't have enough data to know if these same genetic loci affect hippocampal volume. These ones affect or explain hippocampal volume at a level of 2 to 3% in non-European ancestry, so there's obviously work to do. I'll skip quite a bit of this so we can get into some diseases, and we'll talk about some different diseases. I want to just add that there's an important kind of genetic variation that we didn't talk about. So copy number variants or genetic deletions. A deletion can remove some DNA, a duplication can add some DNA. These can cause a huge impact on mental health and neurological disease. Huntington's disease is a disease caused by trinucleotide repeats, CAG repeats in the genome. There are developmental disorders, Fragile X syndrome, Williams syndrome, Turner syndrome, that are resulting from a deletion of genetic material. So this group, ENIGMA CNV, they're making a catalog of all of the common deletions. And when I say common deletions, they're very rare. The most common one, 22q11 deletion, is about 1 in 2,000 people, or 1 in 4,000 people, thereabouts. So you need an international study to find people that carry these. But there's some progress. So the 22q deletion is associated with a very different pattern of surface area in the brain, cortical thickness in the brain. It doesn't look like any of the idiopathic diseases that we discussed. But also, the deletion causes a risk for schizophrenia. Does the pattern in 22q look like idiopathic schizophrenia? Well, notice I used a different scale. So this is half a standard deviation of difference in gray matter thickness in schizophrenia. This is a much higher difference in gray matter thickness, actually excess and deficit in 22q. So we're just beginning to understand the connections between a deletion that leads to schizophrenia in many cases and the circuitry that is obviously very different. Okay, so in the last maybe 20 minutes, we'll talk about psychiatry and neurology and not genetics. I'll move on a little bit. Oh, I want to show you this one. So scanners are expensive. Brain MRI is expensive. You can look at the genetics of brain function with EEG, electroencephalography. It's very easy to do this. I am in a hotel having an EEG, not a usual thing. I hope you don't have an EEG while you're here in the hotel. But it can measure the frequency and synchrony and power of brain activity in different frequency bands. And these frequencies are altered to some degree in schizophrenia, depression, and dementia. So you can look at the genetics of EEG, here a study of 8,000 people. And you can do the same thing. You can do genome-wide association analysis, links multiple genes to oscillatory brain activity. Now, if you're a neuroscience student just starting your degree, you're told that the thalamus and the cortex have oscillators. So there's an oscillating communication between the thalamus and the cortex all the time in different frequencies. It was discovered recently that the GABRA2 gene affects beta power in the brain. What does it mean? Beta power, it's the synchronized brain activity in a certain frequency range in the brain. This is a nice gene. It's one of the receptor subunits for GABA, GABA aminobutyric acid, the major inhibitory neurotransmitter in the brain. So we are beginning to find that a particular marker involved in GABA neurotransmission affects 20 hertz oscillatory activity. And it was already known that this genetic marker was associated with alcoholism, epilepsy, but it was unclear why. So you can begin to see, it's early days yet, that if you were to find a gene that affects a certain brain biomarker, you could begin to build a connection with many of these different disorders where you've unearthed the gene and the connection. Okay, this is interesting, epigenetics. So we all know that the brain and the genome interact, but you're born with a genetic code. The genetic code doesn't vary, except rare exceptions throughout your life. But the genome is also methylated. There's histone modification, there's epigenetic changes due to methylation. And Sylvain de Rivière at King's College London did a study looking at the degree of methylation in certain areas and whether it's linked with hippocampal volume. And this is very borderline results, you have to be careful. But it looks like there's some areas of the genome where across many sites, this is Australia, this is imaging genetic studies in Europe, there appears to be emerging evidence of association between these markers and hippocampal volume. Very, very exciting area for the future. Are there genetic variants that affect the rates of brain growth and degeneration, or the speed of brain aging? Well, you could collect, let's do it properly. You could collect 10,000 people who were scanned twice with MRI and measure the speed in 37 cohorts. This is people aged 4 to someone aged 99 was scanned. And you could say there are certain loci in the genome that are linked to the rate of gray matter loss or the rate of volume loss. So this is a variant of GWAS that could be quite interesting for the future. Can we be mathematically more efficient? So those of you who know, does anyone know what this is? Maybe you do know what this is. I don't know. It's a coding machine, and so it's an encryption machine that makes secret codes. And in Britain, during World War II, there was a decoding machine. They were trying to understand how these machines were making codes. And I feel like maybe we're not sometimes understanding the genetic code well enough. This was used to decode messages, secret messages. But the genome is very complicated. I mean, maybe many markers in the genome act together, and maybe many circuits in the brain act together, and there's some sort of complicated link between sets of genomic processes and circuits in the brain. Anyway, that's just to whet your appetite for future mathematics that can be used. All right, let's jump to some diseases. Oh, I have to show you this one. We need to look at some money. What is this? I skipped an important thing. So let's say you collect MRIs from lots of different centers. They don't always look the same. Like my scanner in Los Angeles might look different from the scanner in Izmir that Dr. Gonul is using. So there are deep learning methods that are being used for multi-site MRI harmonization. And this is the principle. There's amazing techniques in mathematics that can change a Monet picture to a photo, or that can change a photo to a Monet picture. And I would encourage you to look at this. It's a very exciting area. Now, this is just for creative reasons. It's artistic reasons. It's fun. But if you were to try this with MRI, there's a lot of benefit in converting an MRI at one center to look like a reference. And some of these techniques are helping us merge the data from different centers. All right, last little bit. Neuroimaging of these disorders. So Professor Murray gave us a very interesting insight into connections between schizophrenia, bipolar, and major depression. Let's have a look at the different imaging findings for these. So in schizophrenia, this is a set of sites that scan patients with schizophrenia and controls. They're all over the world. And it looked like the main structural difference that they were all able to find, not all of them, 25 of 33 cohorts, was in the fusiform gyrus. Maybe not the most famous area in schizophrenia. Maybe you would think the frontal lobe would show more differences, or the hippocampus. But this is about 1/2 of a standard deviation in the volume of this region. But you can begin to see these are cohorts across the world. This one is in, let's see if we can read it, Dublin. This one is in Osaka in Japan. This one is University of Pennsylvania. There's a lot of consistency, but maybe some centers don't see the same thing. Maybe these folks at this Karolinska Institute, they don't have exactly the same findings. So we're beginning to see that in schizophrenia, for example, there's a very large difference in the brain. If you look at the different disorders, here's major depression, bipolar disorder, schizophrenia, a lot of frontal involvement, 22q deletion syndrome, excess and deficit, epilepsy, large frontal involvement. Obsessive compulsive disorder, I'm not showing the thalamus, but the thalamus is involved. Autism, excess of gray matter, at least in childhood. Alcohol use, a lot of damage to the frontal lobe in alcohol use. And so these are only structural maps. No doubt functionally there's all sorts of interesting things going on that we missed with the structural maps. But notice how different the disorders are. In major depression, actually this is probably not the famous region, it's the limbic system in depression. Bipolar disorder, the frontal areas that are involved in inhibitory control are more impaired, at least structurally. Major depression, not so much. In schizophrenia, huge difference. I turned up the scale so that these are actually small differences, maybe 5%, but bigger than bipolar and major depression. And so you could make comparisons. You could say, if I have a patient who's depressed, or I have a patient with bipolar disorder, is there a difference in the circuitry? It looks like the cingulate area, the limbic system, and some frontal areas show structural deficits in major depression. This is an average of 10,000 people's data. Whereas in bipolar disorder, there's a frontal deficit. The inhibition may be involved in other things. Working memory learning is showing a deficit. So there's a difference in the pattern. But we have to be careful. So one of the things that Dr. Stephen Laurie said yesterday morning is medication use and clinical severity are often confounded. So let's look at 2,000 major depression patients who are taking antidepressants, and we'll compare them with 5,000 healthy controls. There's less gray matter. Let's look at 1,000 antidepressant-free major depression patients and compare them with healthy controls. Well, there's no difference at all. Why do you think that is? Well, the people who are taking the most medication and for the longest time and in the highest doses tend to be more severely ill. Dr. Laurie mentioned this yesterday. So these epidemiological studies that ENIGMA does, they're sometimes a bit misleading about medication. I mean, we don't prescribe medication randomly. So if you ever see a structural MRI map that shows an effect of medication, be a little bit careful. I mean, maybe if it wasn't a randomized clinical trial, the people that were getting the medication were much more ill than the people that were not getting it. Not always. And so we have to be a little bit careful. I mean, a bad interpretation of this would be, oh, well, maybe the medication is associated with gray matter deficit. It's not. It's different people in each case. So there are signatures subcortically as well for the different disorders, schizophrenia, bipolar. You'll see in children with ADHD, these are younger, these are adults. There's a pattern where maybe not so much the hippocampus. There's the hippocampus in ADHD, which is in purple. The hippocampus is not very different in ADHD, very different in schizophrenia, somewhat less in bipolar, somewhat less in MDD. But in ADHD, there are other regions that show bigger differences, the caudate and putamen, which again are involved in, as you know, motor learning and learning of many skills that become unconscious over the course of time. How do these results look if you go to a different country? So Professor Murray mentioned yesterday about cannabis use in different countries. A fascinating talk about Spain, Italy, Trinidad, London, and cannabis use in psychosis. If you were to go to Japan, and this is a wonderful study, this gentleman is called Ryota Hashimoto, and he leads ENIGMA Japan. And if you ever get the chance to go to Japan, I don't know if you know anything about this area of Japan, it's in the western tip. They live among the longest of anybody in the world. Their life expectancy, lifespan is maybe 80, 85, and that's the average. And maybe the diet is different, maybe the lifestyle, maybe there's less stress, there's less drug use, maybe it's genetics, maybe it's a mixture of everything. But what he found was that if you were to study mental illness, in this case brain imaging in schizophrenia, all of the same brain regions come up. I mean, he's looking here at the differences. Maybe I have a better picture. I'll show you it in a minute. All of the same brain regions come up for his patients and for ours. There doesn't seem to be a drastic difference. Now, we mentioned that the environment is not the same, but maybe the imaging, we can look into some subtle differences between there and here and find out if there's anything that's advantageous for them. Linking MRI brain alterations to cell-specific gene expression. Some people say, well, here's just a picture of the brain. Can we say anything about the cellular differences? So Thomas Paus, very well-known neuroscientist, has been interested in correlating human brain gene expression with these deficit patterns. And there's certain genes that are markers of certain cells, not exclusively, but they're good tags for cells. So he's trying to look into, in its early days yet, whether certain cell types have preferential abnormalities. And this is a very, very interesting line of work called virtual histology. You can do meta-analysis of other kinds of brain maps. So this is voxel-based morphometry. It's a group of patients with obsessive-compulsive disorder. And it's a meta-analysis of 20, 21, 22 studies. And you can see these maps are being built up. You'll notice that there's a lot involved. Often with a small study, we tend to pick up one region, maybe in the thalamus in OCD. But as you begin to look in larger samples with higher power, you begin to see that, in the end, a lot of the brain, or nearly all of the brain, is involved. Why are some of these studies disagreeing with each other? So This group in Italy, Fabrizio and Federica Piras and Gianfranco Spalletta, they were interested in diffusion imaging. It looks at microstructure in children and adolescents with OCD. And they found that 3 factors influence what you find. And so you can certainly find abnormalities in microstructure in OCD, but some people don't find them. I mean, why does one group find them and another group doesn't find them? They found that the duration of illness, the age of onset, and the percent of the cohort that has received medication. In OCD, there's some people are medicated, some are not. The effects depend strongly on the duration of illness, longer is worse. The age of onset, earlier is worse. And the percent of the cohort medicated. Now, I want you to remember one thing. More medicated was poorer microstructure. It doesn't mean the medication is bad, it's just associated with having a more severe illness. And so, just to remember this, each dot here is a cohort. When these patterns don't appear to be replicated, it's not always the same people. They might have been ill longer, medicated more, or get the disorder younger. The biggest effect in psychiatry that we've seen in imaging is anorexia. So this is actually a positive story in the end. Esther Walton and Stefan Ehrlich at University of Bath and University of Dresden found that there's a 1 standard deviation lower gray matter thickness. On average, people are very different. In eating disorders, in particular anorexia nervosa. But after they receive treatment, after they have nutritive restoration, I mean, they're helped cognitively and also are treated clinically, these deficits normalize. So we have to not think, Professor Murray mentioned this yesterday, we have to not think of these disorders as degenerative and necessarily progressive. Even in an eating disorder where there's malnutrition of the brain, they have found in their very interesting work that the brain can be restored. This is a really famous one. So there's probably some of us in this room that know, I think Professor Murray, you know Tim Crow, you know Eve Johnston. So they always said, well, they didn't always say this, but they were one of the first to say that maybe in schizophrenia, the lateralization or the asymmetry of the brain is altered. Do we believe it? Some people believe it, some of us don't believe it. So there was a very nice study, a partnership between the ENIGMA schizophrenia group and the so-called ENIGMA laterality group. They look at structural asymmetry of the brain. And Tim Crow and Eve Johnston said, we think that, well, they did the first CT scan in 1976 of people with schizophrenia, and they noticed the enlargement of the cerebral ventricles. Eve Johnston wrote a very interesting paper in The Lancet in 1976 to show structural differences in schizophrenia. A little later, they said maybe there's an imbalance of the hemispheres. This was recently resolved in the affirmative by Dick Shiven. There is an altered asymmetry, on average, maybe not everybody, in schizophrenia, but the effect size is 0.05 of a standard deviation. So there's a reduced asymmetry, structural asymmetry, in the group of patients with schizophrenia. And to be honest with you, it required about 10,000 people to confirm this. But I think that's a nice piece of information on a long-standing hypothesis that's been going on for nearly 40 years. I want to just say about meta-analysis. I mean, we all act like meta-analysis is a good idea. Some of these diseases are progressive. So in Parkinson's disease, there are 5 stages, beginning with tremor, motor rigidity, and in the end stage of Parkinson's disease, these stages, stage 4 and 5, a patient will be confined to bed. They'll have very little speech. They'll have very little movement. So it's important to recognize that we should move beyond meta-analysis. I mean, there'd be no such thing as an average brain in Parkinson's disease. This axis is the progression of Parkinson's disease. And as we said, there are other dimensions, the age of onset, the duration of illness, and the type and duration of medication. So we have to think a little bit about differences as well as averages. But they can be used for staging. So these brain maps are actually very useful for looking at the stages of Parkinson's disease. These are the different regions of the basal ganglia. And from early to late Parkinson's disease, you'll see a progressive difference in the structures. Ataxia is a nice, and people that look at ataxia, it's a motor disorder, they don't look at the cerebrum very much, but they look at the cerebellum, and that's a flat picture of the cerebellum. Ian Harding, who leads this, says, in ataxia, the deficits are much greater if someone has an early onset, much milder if the person has later onset. But then, depending on the age of the patient and the duration of the disease, there's also an axis where there's obviously greater deficits the longer you've been ill, or greater deficits the earlier you were diagnosed. So you'll begin to see across neurology and psychiatry there are many consistent patterns about duration of illness and age of onset. And they have a very nice toolbox. If anyone's interested in analyzing the cerebellum, they have a very nice toolbox to do it. Clinical high risk. So a very nice talk. Professor Murray, Professor Laurie have talked about association of structural brain imaging measures with psychosis onset in people at high risk. So this is Maria Jalbo-Zakowski and Dennis Hernaus. They've been looking at which brain regions, either individually or as a group, or Stephen Laurie mentioned a very nice cross-modality study that predicts psychosis. And they're beginning to see that there is a link between certain areas, sometimes not always, we would have thought of these in advance, and future onset of psychosis. And Professor Laurie gave a very nice talk about care in using these, how we should be careful with patients in thinking that this is, fate, or that there might be some other factors, including good treatment, that can reverse or inhibit it. I'm going to skip this one and we'll cut to the end. So brain mapping is a lot of fun. I mean, we talked about brain structure, but you can map the connections of the brain with diffusion tensor imaging. Here's the neural pathways. Everything I've talked about today could be done on this too. So you could say, here's a brain, we map the tracts, there's the cingulate gyrus, the frontal cortex, here's the temporal lobe. It's really beautiful. And you could say, well, can you tell me anything about the genetic factors, the environmental factors, the medication factors that alter the synchrony? So Professor Ali yesterday was talking about functional synchrony in these circuits and how in major depression there are shifts among the systems that are active. So you can represent the brain's connectivity by looking at the structure, looking at the connections, making a matrix of connections between the different parts of the brain. And in here you can measure whether there is a connection, what is its integrity, its microstructure, or even its synchrony. You can look at its functional synchrony. And this makes an image. And so everything we talked about, you could put those images into a statistical pipeline and try and understand factors that influence not just differences in a population, but changes related to treatment or related to positive outcomes and other things. All right. Some lessons we learned. Okay, so this is the last bit, last 2 minutes. I think global participation is very important for science. And so ENIGMA, I mean, we're all sitting here thinking about this, but these are activities that are in 45 countries. It's politically very beneficial when projects are led from different countries. I mean, sometimes people think they know all the answers, but you get a different perspective. People are seeing patients every day. You'll hear about big data, greater power, more efficient, all the computers. But all the computers in the world are no substitute for your minds. I mean, you have individual experiences every day where you're gathering expertise. So at a conference like this, hearing from you and hearing from your own individual experiences is probably more important than all of the data. I mean, the data doesn't do anything on its own. So, I mean, we've had a very wonderful experience. I mean, this was yesterday here where Professor Ali Safet Gurnul leads this lab in Izmir. So they're collecting, this is very nice people I met earlier this week, they're collecting resting state, other functional task-related MRI on people with schizophrenia, people with major depression. Dr. Aslihan Demir is the co-author on the study, one of them we've been talking about on suicidal thoughts and behavior. And there was a really nice presentation from one of the medical folks at this lab earlier this week about some of the studies we've talked about today. So I mean, this is a great effort. What are we going to do with geopolitics? So there was it's very sad, actually I shouldn't make light of it. There was a very active ENIGMA effort in Russia, in Novosibirsk. My colleague Vladimir Zelman set up a network of sites across Russia. And they study PTSD, they study schizophrenia, depression in Moscow, Novosibirsk, across Russia. In Thailand, there's a very active group, the ENIGMA Cipher Project, studying little children with HIV. In Bangkok, about 10% of infants have maternal HIV, and they say, well, if you really want to study risk in Thailand for altered brain development, you should look at infectious disease. Infectious disease is very, very common, and these folks in Bangkok are scanning little babies with HIV. So we shouldn't lose sight that there's also infections that alter the brain. In India, there's a very nice group in India, in Bangalore, that are scanning people. They want to know, are the statistical norms that we use in Western Europe, in the US, in the Middle East, do they make sense for people in India? Maybe they do, maybe there's some differences. And we talked about in Japan, lots of partnerships trying to replicate, I shouldn't say replicate, I mean to understand what the modulators are in different countries of the brain patterns that are found in different disorders. Very active brain mapping going on in Iran. So until very recently, it was not allowed for a researcher from the US to work from people in Iran. But in Turkey you can. In Turkey you can work with folks that are scanning people every day at the center in Tehran. Also a nice center in Isfahan is collecting data. They have a network of 6 centers collecting neuroimaging data right now from people with different psychiatric conditions. So something interesting about Iran and the Middle East, there's not a great deal of drug and alcohol use. And so Professor Murray mentioned this. Maybe in populations, I mean, people are different in many ways, but in populations where drug use is very, very low, and maybe in Turkey to some extent, drug and alcohol use is much lower than where I grew up in the UK, and to some extent in the US, we'll understand some of the determinants of mental health. And what about socioeconomic status? If you're this chap here, you probably have access to healthcare. I would bet that, and me, I mean, I'm very lucky. I live near a hospital. So we can't forget that, people's access to resources, expertise, it might matter a lot. We didn't measure that in any of the studies, and so that would be important to bear in mind. Just to wrap up, next year we'll be doing harmonized analysis in ENIGMA of diffusion tensor imaging, microstructure in 10 different disorders. I didn't talk about AI and machine learning and deep learning, but many of you that are talented mathematicians will have a lot to offer. In genomics, do different subtypes or dimensions of diseases have different genomic drivers? I mean, there's a lot of interest in the heterogeneity. And a lot of this is being done through partnerships with ENIGMA, Japan, India, a new project in Pakistan, and China to look at the social and cultural, and certainly the environmental effects on brain health that you can get at through these. There's many ways to do it, but one of them is through geocoding, where you label where the person was scanned and try and link that with urban density, access to resources, access to healthcare. So if you're interested in ENIGMA, we have a YouTube channel. You can go and have a look. Here's Odile van den Heuvel, Barbara Franke, the geneticists working on these projects. You can listen to them. They do a better job than me. And you can take part in ENIGMA. You can go to the website, go and have a look if there's anything, maybe join some of the phone calls if you like meeting people. There's different disorders being studied. And you can join. I mean, you don't have to participate. You can just listen in and see what people are doing. It's very open. Anyone can join this. And just a really big thank you. I mean, there's been people from all over the world, here in the Netherlands, people in China contributing, people in South Korea, and as I said, Dr. Ali Safet-Gurnal and your group here has made really important contributions to these studies. It's actually funded by people all over the world. So I live in the US, so the NIH funds my group. But I mean, think of all of the agencies throughout the world, private, and state and federal funders that have sponsored the collection of the scans and the evaluation of the cohorts. You're always welcome to come and see us. I can't guarantee that air quality in Los Angeles is very good, but it's very pretty. You can come and see us at the ENIGMA Center in Marina del Rey. And just a final lesson. So this person, he was very wise, and he wrote in ancient Greek, and he said, individually we contribute little to the quest for truth, but by working together, the whole vast world of science is within our reach. It's very, very, very nice sentiment, and here it is in his metaphysics, which is, and so I just wanted to say, Budesi Tukche Sheviren Teshumana Teshikula to you for, in the translation room for doing such a wonderful job today, translating between Turkish and English. Thank you very much.